Um, so yes, my name is Kate. I'm from Fred Hutch Cancer Center, and today I'll be talking to you about data visualization beyond the basics. So for our outline today, oh, also that link and that QR code you can use to actually have the slides with you and access them. So our outline for today is that we're first going to talk about establishing best practices for data visualization. What are the types of data that you're going to be working with? How do you choose plots that you're going to use based on the data? And how do you present complex data with clarity while making your visualizations accessible and following ethical practices? And then for the second half of the talk, we'll explore creative or non-conventional visualization approaches. So first, for establishing best practices, let's first overview the types of data visualizations there are. And there are two types of data visualizations. The first are exploratory, and these are visualizations that are not meant to be shared widely. You're going to make them quickly, and you're just exploring your data. You might be looking for something specific, but this is not something you're going to spend a lot of time polishing. This is for you to use in your labs when you're comparing data with each other or looking at it yourself. You might want to see if you have some missing data and possibly patterns causing that missing data. You might want to see if some variables are correlated or have interesting patterns or distributions to them. You might want to see how your non-numerical data is coded within the data set. Are there some cleaning steps or synchronization steps that need to happen? This example right here is using a package called Nanier in R to look at the Palmer Penguins dataset for missing data. And so in black on this heat map, it highlights when there's missing data and which variables or columns have missing data. And in gray, it's saying the data is there, it's not missing. And you might notice that the comment section has a lot of missing data, but that it doesn't tend to co-occur with missing data in other columns, suggesting that they're using the comments to explain why they might have missing data. And so exploratory visualizations are not something you're polishing, not something you're sharing. And there's certain types of plots that just work better for exploratory visualizations than the second type, which is expository visualizations. These are the ones that you're talking about, and we're going to spend most of our time talking about and discussing are the ones that you want to share widely with others. You're going to use them to communicate or explain a specific point. They're going to be polished. You're going to spend a lot of time making them pretty and making sure that other people can clearly understand what you're trying to communicate. And it's often an iterative process because you're trying out something and you're going to see how it looks, and then you're going to improve it. And you might do this several times until you have a solid visualization that you confidently want to share with others. And so examples of expository visualizations are plots on a poster, tables in a paper, and visualizations within a talk or presentation. And yes, tables can still be a type of visualization and are a very strong thing to use sometimes. I linked a blog here about building up a ggplot2 figure, and it talks about the steps in that iterative process of turning your visualization into an expository, shareable, very polished visualization. So the types of data that you're going to be using are normally numerical data, especially with the work that you all do, where you're going to have some sort of quantity that represents a count or a measurement, and you can do mathematical operations on these. There are two types of numerical data. The first are discrete, whole numbers, and the second, continuous or real numbers, those numbers that have decimals, so like a temperature, 98.6. I was sent some papers by, I believe, Omkar before this presentation from papers from your group. And so I looked through there and found some examples of numerical types of data in those papers. And these include blood test result values, a patient's age, the Breslow thickness of a tumor, the length of stay that a patient might have, the survival probability of a population, or also the creatinine clearance rate for kidney function. The second type of data that you're going to often be working with is categorical data. And this qualitatively characterizes, categorizes, or groups individuals with labels. It may look numerical, like an ID or a ranking. A common example of this is 1, 2, 3, but you can replace 1, 2, 3 with ABC. The 1, 2, 3 isn't a necessity, isn't a requirement. It's just a way of grouping. You might also define your categorical data based off of numerical cutoffs. So if we consider those blood test results, you probably have numerical cutoffs to say this is lower than average, this is within the normal range, or this is a high value. You can sometimes do math with categorical data, but it might be awkward or not possible. Rankings are really the best example I have for doing math with categorical data, where you might try to find an average rank, and that's pretty well accepted in the field, but typically you're not going to do any type of math with categorical data. And there are two types of categorical data. The first is nominal, that can't be ordered. So this is something like your case and control categories, or maybe the categories of different types of medication or treatments. The second is ordinal. This can be ordered, something like ABC, something like never having a symptom all the way to always having a symptom. You can order those. There's an accepted order that people understand. So looking through the papers from the group, some examples of categorical data that I found were treatment group, the sex of a patient, the Charlson-Deyo Comorbidity Index, which this is an example where you are doing mathematical operations, adding together those index values to get a final index score. But it's really a categorical variable because it's describing groups. The tumor location, the ECOG performance status, and surgery receipt for a patient. So when you're choosing a plot type given your data, you want to keep in mind, what data do you have? Is it numerical? Is it categorical? Is it a mixture? Because this will greatly impact the types of plots you can use. There are certain plots that need numerical data. You also want to think about how many variables do you need to visualize. Certain plots have a minimum number of variables that they need. You also want to take into consideration, what is the goal of your visualization? I have 4 main goals here. This is not like fully extensive. There can be other goals, but you might want to look at a distribution of values. You might want to show a correlation between 2 variables. You might want to find a ranking. You could also be interested in the change over time or after an intervention. Here are 4 common plots that you're probably familiar with, and when they're appropriate, given those questions we just discussed. With histograms, you're using numerical data. You're going to have one variable unless you're looking at a few variables and each has its own histogram. And its goal is to look at a distribution. While with a scatter plot, you have numerical data and you need at least 2 variables, one for the x-axis and one for the y-axis. However, you could have an additional variable that might be categorical that affects the color or the shape of those points in the scatter plot. And this is often going to be used to show a correlation. For a bar plot, you're likely going to have a mixture of numerical and categorical data. And you're going to have at least 2 variables, one for the categories along the x-axis, in this example here, and one for the values or the height of the bars, which is numerical data on the y-axis. And bar plots are often used to find a ranking across the categories. And a line chart, similar to a scatter plot, is going to need at least 2 variables with numerical data for the x and the y-axis, with perhaps categorical data and another variable to affect the color or the shape of the lines. And it's going to look at change over time. So this is just a glimpse with some plots that we're hopefully comfortable with of how those questions affect the plot we're choosing. There are a lot of plots, and this example on the slides is a website that gives a breakdown of the questions that you might be asking and the type of data you might have and what plots there are. And you can click on the type of plot to learn more information about it. And so it's a really great resource. It is not exhaustive. It's even missing some of the plots that we're going to discuss today, but it's a great place to start. And it often gives suggestions. When you're choosing a plot, another important consideration is that there are often standard or typical plots in the field, and they're often complex and there's no getting around that. It just is the plot that people use. So this is more common in genomics and proteomics, but in biomedical research, including clinical work, some of these are becoming commonplace. And so two examples that I have here are first on the left, chromothripsis and a Circos plot. So chromothripsis is when the chromosomes have some catastrophic event and they break apart. And so they put themselves together, but often not in the correct way, and they'll combine chromosomes. And so a Circos plot is being used to show which structural variants happened when chromosomes put themselves back together and joined with a different chromosome. And so that's the inner layer. And this Circos plot is even more complex because it's showing things like somatic variants on the outer layer. So it is labeling with each layer what you might want to look at, but this is a very complex plot, but it's also the plot that the field uses. On the right, I'm showing a second complex plot, protein-protein interaction networks. Very, very common and very, very accepted that you want to see what proteins are interacting with what other proteins. In clinical research, there are additionally more standard typical plots. These are less complex to visually inspect than the ones that we just looked at, but they're pretty complex to prepare the data. And if you're new to looking at these plots and you're not used to looking at them, they have a bit of complexity or a bit of extra context that's necessary to understand them. So it can be a lot for new viewers. An additional area of complexity for these is that not all software can plot these. So GraphPad Prism is going to be able to plot a Kaplan-Meier curve, no problem, but other software, you might have to figure out how to plot it, given that it's not an everyday, every single field kind of plot. On the left, I'm showing a Kaplan-Meier survival curve, which is just a line plot, but has some differences that make it a step. And on the right, I'm showing a waterfall plot, which is very common for looking at how tumors react to treatment and compares to a baseline and specifically looks at like long-term patient response. A third one that I'm not showing an example of, but we'll look at later, are visualizations that explain study design or participant trajectories throughout. Those are very complex as well and have the same considerations as these and are just standard typical plots in clinical research. So given that you're working with complex data and sometimes you're stuck with plots that are just complex, here are some tips to reduce the complexity when you can, when you're making a plot. And my first tip is that you want to use your visualization or your plot to answer only one question. If you have an additional question, add a second plot that works together with the first plot or is completely separate if it's a completely separate question. Don't try to fit too much information and too many answers on one plot. A second suggestion is to add annotations or labels, such as dashed lines, names, colors, anything that can help add some clarity to what you're showing and might show different groups or might show a baseline and defines important information. Now, while I think it's really important to add these annotations or labels, you want to make sure to not overclutter your plot and have everything just on top of each other. You also want to minimize white space, so there's not too much white space with nothing going on. In addition, whenever you add a label, especially an axis label or labeling a line or a data point, use clear labeling and names. Make it readable, extra large text, and make it understandable. Make it something that is avoiding jargon, avoiding acronyms unless they're defined somewhere, and is something that someone else can understand quickly without having to have it explained for like 10 minutes. An additional way to reduce complexity is using facets or subplots. And so maybe you have several categories and they're all sort of on top of each other. You can use a subplot or a facet to break those categories apart and look at them next to each other or in different subplots. Another really good tip that we'll see a couple of examples of are arranging your data according to the typical reading order. We typically read left to right and top to bottom. And so when someone is looking at your plot, that is how they're going to typically look at it. And so you want them to go through the plot starting left to right and top to bottom and not having to look somewhere else first to understand it. I said that we wanted to talk about making our visualizations accessible and doing that ethically. And so what does ethics have to do with data visualization? First, with the work you're doing, you probably have some identifiable data. And I'm we always say this in trainings and discussions, make sure not to include identifiable data. That is one of the basic steps for ethically working with human and clinical data. Another large part of ethical visualizations is clearly and accurately communicating. You want to avoid misleading messages. And if someone is just looking at your graph briefly, you want to make sure that there's not some unintended message that comes across. You want to compare groups fairly, and we'll see a couple of examples of this. We want to include all relevant data or subgroups. And if a sample gets removed, describe the removed samples. Don't just let them fall out or not be there without describing why or showing the removal. I think that we'll see an example with that with Sankey diagrams later. Ethical visualizations are going to improve the viewer's understanding rather than confuse or mislead them. So rule number one for ethical visualizations are using fair axis limits rather than truncating them. So in this example on the left, we're not really comparing these groups fairly because we're making it look like there's a larger difference between the pink group and the other groups because we've truncated the axis. When we use the axis on the right, we see that they are not that far apart from each other. They're quite similar. There might be too much white space up here for the axis limit, but perhaps you have another group that goes even higher. Rule number two is ordering your axes in a logical way that corresponds to the plot's message. So in this plot, it's trying to plot weight loss throughout the month of June. But this graph, when you look at it, it looks as if weight is increasing across the month. And that's because this y-axis over here starts at 280 and goes to 270. Well, if you reverse that and put 270 and go to 280, then we would see weight loss happening throughout the month of June. So axes in the wrong direction can be misleading. And if you look at this briefly, you'll walk away with the wrong message. You also want to make sure that the visualization accurately represents the data. So on the left, we see a pie chart where 45% is the larger area and 55% is the smaller area. That's likely a mistake. It should be 75% to 25%. And mistakes happen. Code has bugs. Something might have gotten typed in incorrectly. So always verify. In woodworking or any type of home maintenance, people say measure twice, cut once. More like check twice before sharing or publishing. Make sure that your data makes sense when you look at it within the visualization. You want to use clear and accurate axis labels. On the left, I have no idea what this is plotting. There are no labels here. I see that the purple group presumably has a higher something than the green group, but I don't know what. Well, on the right, we've added axis labels and we've made it a large text so that people can see that we're comparing gender groups and we're comparing height for those groups. The final rule for ethical visualizations is making sure not to mislead and separating out those plot elements. So this plot is looking at iris petal characteristics for 3 different species for the width and the length of those petals. This is kind of cool because it looks like some sort of like exclamation point, but you can't find anything informative from this plot. And it might even look like the species don't have differences because all the data is right on top of each other. So what can you do to separate out the plot elements and verify whether the species have differences or not? You can adjust the shape, size, or transparency of these points. You might, in some cases, need to log scale or transform your data to separate out things. And it might be helpful at sometimes to use a zoomed in set that if you need a large scale of data and one part is very, very clumped, you focus in on that for just part of it and zoom that part and then look at that clumped part while still looking at the whole thing together. Another one that I don't have listed here is subplots. Maybe you want to plot each species in its own subplot or facet it. So we discussed why our data visualizations need to be ethical. Why do they need to be accessible? And this comes down to who your audience is. There are a wide range of audiences, and you might have audiences with visual impairments, whether that be low vision or blindness or color vision deficiency. They might have motor impairments, which is going to be mostly relevant if you have some sort of interactive visualization. You might have audiences or audience members with dyslexia who are prone to distraction. And very commonly, you're going to have audiences who are unfamiliar with your research. In this talk, we will mostly be discussing visual impairments and audiences unfamiliar with your research. If you are interested in any more of the information, check out the linked blogs. One of the easiest, best steps you can do to help make your visualizations accessible for these audiences is using color vision impairment friendly palettes. So on the left, I'm showing a simulator that shows colors in a color palette in the true column. And so I have 6 colors here, and this is called the Viridis palette. And for each row, it's corresponding that color to 3 different kinds of color vision impairment. And it does that for each color in the palette. So if you look within a column, you can see that the colors are still distinguishable for that color vision impairment, that they can still see the differences between those groups or for those colors. Viridis is a fairly simple to use palette with most visualization software pretty directly supporting it. There are other palettes that are designed for this purpose, just like Viridis, and they're listed in the simulator link that's provided below. Perhaps you have very specific colors in mind. You can check your color choices for accessibility, either using that simulator that we just looked at, a contrast checker, or a screen overlay simulator that will show you what the colors look like. Other best practices for making accessible visualizations include not relying on color choices alone to make distinctions. Use shapes, patterns, or direct annotations too. Not every single visualization that you make needs to have that, because that can also be repetitive, and you often want to avoid repetition to make your plots simpler. But if you're using a color palette or color choices that you're not 100% sure are going to be distinguishable, it is very good practice to use shapes as well as colors or some other repetitive method so that it's not only color to make that distinction. And we've already discussed this, but using clear access labels, larger text sizes, avoiding jargon and acronyms unless you've defined them, perhaps in a caption. And using a sans serif font, this is very good for people with dyslexia. It's also very good practice when you're making an expository plot that you want to provide alt text, descriptive labels, a title, and a caption. So if you're putting this plot on a slide and you're going to talk through it in a presentation, you're probably not including a caption. You probably want to put the caption in your speaker notes. You'll definitely have the caption if it is in a paper. But the whole idea of this is that your plot should stand alone. If someone is looking at it without you there to explain it, they're going to be able to understand what you're trying to communicate to them because they will have all of that information, the descriptive labels, the title, the caption. Also, separate out your plot elements. Don't overclutter the plots. Like we discussed, if your points are on top of each other, figure out how to have some distinction between them. And use a layout that follows the typical reading order, left to right, top to bottom. All of these help make your plots more accessible to viewers. So the takeaways from this section: carefully inspect your visualization to make sure it matches your data and expectations. Is any data missing? Spot check some values compared to your data. Put the plot and your data side by side. If you have lots of data, pick a few random subsamples to check. And if you look at the plot quickly, is it misleading at a glance? Consider that weight loss example we looked at. Can someone who didn't help make the plot or doesn't know how it's supposed to look understand what it's meant to communicate? Because your plot should stand alone. So that is a great test. Give your plot to a friend. Have someone else look at it who isn't familiar with the work and see if they understand it or areas where you might be able to improve it. So for our second section of the talk, we're going to explore some creative visualization approaches beyond the typical standard plots. And for this section, we'll have a plot. I'll describe the plot. I'll describe when you might want to use it, its strengths, its weaknesses, and some alternatives to it. I'll make an example plot with Palmer Penguin data. This is not necessarily always going to be a good example. We might even talk about the weaknesses of it. And then I'll try to give at least one example of the plot from papers. For the first set of plots, these are ranking and distribution plots, such as a violin plot. So a violin plot is going to display your data much like a histogram, but specifically it's displaying densities of the data. And so narrower sections correspond to little density, while wider sections correspond to more density in your data. And this is going to be used for numerical data with it likely one variable. You might have multiple variables if you're looking at multiple distributions, which also means that you probably have some categorical data there. And it's best to use this with a minimal number of categories. The more violin plots you have, the harder it's going to be to distinguish them. And this is mostly used for showing distributions and ranking groups. The strengths of violin plots include that they're much better at comparing multiple distributions than histograms. Additionally, it's allowing you to look at the distribution shape. Is it skewed one way or the other? Do you have multiple modes? Then something like a box plot is going to allow you to do. There are several weaknesses to violin plots, though. They don't show you the individual data points. You're seeing the density of the data points, but you're not seeing the data itself. And it's going to have limited usefulness when there's little amounts of data. Similarly to how you can't see the data points, you might have categories that have different sizes of the categories, and that's going to be difficult. There's no way to see that. The method for creating the density visualization is also a bit arbitrary, so that can be a weakness. Alternatives to a violin plot include using a box plot, and you can do something called jitter that adds the individual data points. Also ridgeline and rain cloud plots, which we'll discuss shortly. This is a violin plot example with the penguins. So the penguins, there are 3 species, Adelie, Chinstrap, and Gentoo. And I'm plotting the flipper length across these species for the female and male sex. And I have female in the dark purple and male in this green. And we see that the Gentoo species has the longest flipper length and that males tend to have longer flipper lengths than females. We might see some areas of multiple modality, like I was discussing, with specifically this Gentoo female population. There seem to be possibly 2 modes. For the most part, I don't see much skew, but this is the strength of violin plots. You're able to look at those distributions. A note on the color palette you just saw. We're using a greenish color, and you might have heard that if you want your visualizations to be color blindness friendly, not to use green. And that is because you might not want to use red with green so that it's friendly for people with red-green color blindness. And in this case, that greenish color is distinguishable from the other color that we're using. So it is accessible. I've made a box plot with jitter points example for that violin plot we just saw. We have the species still here, and the boxes are colored differentially based off of the sex group, with the flipper length on the y-axis. And for the most part, we see what we saw with the violin plot, specifically that the Gentoo penguins have that higher flipper length and that the male populations tend to have that higher median or the higher flipper lengths than the female populations of each species. It's a little bit more difficult to see something like the multiple modality that I saw with the violin plot specifically here, but this does correct a few of the weaknesses of the violin plot because you're able to see the data points. You're able to see how many data points there are for each group. So another alternative to violin plots are ridgeline plots. And these are going to sort of look like the mountain ridges or a mountain range or a few mountain ranges all near each other. And this is showing the distribution of multiple categories of numerical data using densities, half of a violin. You're going to use this with numerical data, categorical for your groups. You'll have at least 2 variables because you're going to have at least 2 mountain ranges. And you can use this with a much higher number of categories than you can a violin plot. In fact, that's what it's designed for. It's going to show distributions and ranking groups, just like violin plots. The strength of these is that they're efficiently displaying distributions for many groups of categories. And they're showing that distribution shape skew, multiple modality, just like violin plots. The weakness is that they can be cluttered if too much overlap is happening between groups or there's not a clear order. These work best when your data is well behaved and there is separation among the groups. It's good for general comparison among groups, but if you want to find precise quantitative differences, this is not your answer. You might want to use a table if you need to know some summary stats for that many number of groups. The method for creating the density visualization is a bit arbitrary, just like violin plots, because it's using violin plots, so there is a way to bin these like histograms. The alternatives for a ridgeline plot are the box plot with jitter, which we looked at a couple of minutes ago, a strip plot, which is just showing the data points and nothing else, and they're just in a strip. And in some cases, you might be able to use a sorted heat map that is showing the groups and you've sorted them to be in the order that you want them to be. And we'll look at a heat map a little later. For this ridgeline plot example, this is the same plot that we looked at as a violin plot and a box plot, but now it's a ridgeline plot. So on the x-axis, we now have flipper length instead of on the y-axis. And on the y-axis, I have those different groups where it's the species and sex combo. So I have Adelian female, Adelian male, chinstrap and female, so on and so forth. And we see that general thing that we saw before of the Gen 2 male being larger than everyone else and the males having slightly more distributions than the females. I don't really see the Gen 2 female multimodality anymore. And that could be due to the density arbitrariness that we talked about. Or maybe there's just like a slight area here and the modes are just very similar to each other. There might be like two shoulders there. But the strength of the ridgeline plot example is that you can do even more groups. And so with this example, I've now increased the number of groups that we are comparing. And I have further added groups that each species might have an island associated with it. So the Adelie penguins can be found on 3 different islands, Biscoe, Dream, or Torgerson. Chinstrap is found only on the Dream Island, and Gen 2 is only found on the Biscoe Island. So we have even more groups by looking at the species, island, and sex together. And so they're meant to be used with more groups like this, and this works pretty well because there's a nice progression. So rain cloud plots, what are they? They're combining the 3 types of plots that we have just looked at. They're combining density plots, so that ridge. They're combining box plots, which show you those summary stats of the median and the quartiles, and they're showing you the individual data points. And so this ends up looking like a raining cloud. And you're going to use this for numerical data. You'll have categories for groups, and you have at least one variable. You might have a few. It's going to be best, though, with a minimal number of categories. Unlike ridgeline plots, these do not scale well to multiple groups. And it's showing the distributions and ranking groups, just like the ones before this. Another note is that rain cloud plots are very good for exploratory because they're showing you everything. And so it's giving you all the info and it's showing you all the things that the other ones are lacking. And so given that it might be cluttered if you have more than a few groups or a lot is going on, this is going to be best for you looking at and exploring and not necessarily widely sharing. Alternatives to it include violin plots, box plots with jitter, or swarm plots. And swarm plots also show individual groups, individual points, but instead of putting them in a strip or a line, it shows them sort of like a beehive or a bee swarm. So in this rain cloud plot example, we have the density, we have the box plot, and we have the individual data points. It sort of looks like a raining cloud. I've reduced the number of groups down to just 3, where we're just looking at the species, Gen 2, chinstrap, and Adelie, and we've pooled male and female data together. Because when we split those out and we have more groups, this is looking more cluttered. It's more difficult to see those box plots. They're really squished. And so this is the downfall of rain cloud plots. If you have too many groups, it's going to be harder to look at them, and that info that you're adding then becomes difficult to actually look at. So to summarize these plots that help you compare groups, violin is good if you want to look at a distribution and look at it at a glance. Box plot is great if you want to see the data and the general summary stats like the median. Ridgeline is going to be great at comparing the distributions of lots of groups, while a rain cloud is great when you want to go deep and see everything for a small number of groups and just explore. So the next set of plots that we're going to look at are group composition and response plots. The first of these is a stacked bar chart, which is going to display multiple categories within a single bar, either stacked on top of if it's a vertical stacked bar chart, or stacked next to if it's a horizontal stacked bar chart. And you're going to use these with numerical data for the height of the bars and categorical data for the groups. You have at least two variables, but probably more. And you're going to find a ranking or a change over time. It's best to use these if you're comparing subcategories within a bar to consider how subcategory contributions to the total, how they contribute to the total bar, and perhaps how that changes over time or across bars or groups. You want to avoid, though, if you have a lot of subcategories, and we'll see an example of this. You also want to avoid if you have, like, if you want to compare a single subcategory to a different subcategory over time. These are not good for that. So the strengths of them is that they're summarizing the complex group composition in a compact way. And you can compare composition over time or across groups. The weaknesses are that it's going to be difficult to compare subgroups that aren't aligned, those subgroups that are in the middle of the bar. And we'll look at that. If one subgroup contributes too much to the bar, it dwarfs the other subgroups, and it can be difficult to see what they're contributing. And it might look like it's far less than it actually is. Another weakness is that if you're using proportions, you don't see the raw numbers of the data. I personally prefer using proportions on my numerical axis when I work with stacked bar charts. I have made exceptions for this if I only have two subgroups and I can directly add numbers to the plot to my bars. And some people prefer this. But if you have very, very small bars or small numbers in your group, that can be difficult to see. And so stacked bar charts are not always a win. There are alternatives to them, a lollipop plot especially, because it removes the general width of the bar. And so you have a little bit more room to plot things next to each other, even on the same line, and just have multiple dots on the same line for a stacked bar chart. Tables can be a really good alternative for stacked bar charts, as can be line charts if you want to look at change over time. And we'll look at waterfall plots later, but if you wanted to just focus on one subcategory, a waterfall plot could be used. For this example with the penguins data, I hypothesize that every island is going to have pretty much 50-50 female and male on it. And so I have an annotation for 50%, and I'm looking at the percentage of sex on each island. And sure enough, each island appears to have nearly 50-50. It's not exact, but you wouldn't expect it to be exact. And so this is a strong bar chart if that is what I'm trying to communicate, that each island has about 50-50. It's going to be not as strong of a bar chart if I'm trying to communicate something else. Well, this stacked bar chart, it's too complicated and the text is really small. And even if I tried to break down what it's trying to communicate, it's going to take forever and it's going to be difficult to understand. So this would be a good example of where it might be better to split the bars out so they're next to each other, or using an alternative means that isn't a stacked bar chart. For this stacked bar chart, this is a graph that I made in a rotation project presentation when I was in grad school. And this slide was hidden for that presentation. I didn't actually present it because it has a major weakness. It's showing raw counts. And that's a problem because of what it's doing. It's looking for structural variation identified in normal tissue, tumor tissue, or if that structural variation was found in both. And the expectation for this is that normal tissue is not going to have a lot of structural variation that's unique to it that tumor tissue doesn't have. Tumor tissue should have more structural variation that's unique to it. And so I'm likely to find structural variation that is shared between the both of them. And so my furthest over bar is just so much larger than the other two that it's very difficult to see the subgroup composition of normal only. Looking at this, the little like teal color, the translocations, I can't tell if that's in the normal only or not. However, if I change that to using proportions instead of the raw counts, now I can see that yes, normal only does have translocations. The example here, though, is that I've discussed what if your subgroups aren't aligned, especially those middle groups. Consider the purple or the duplications for the ones that are shared across both and tumor only. These are not aligned, and I can clearly see that normal only has a larger proportion of duplications than the other two. But it's difficult to compare these two because they're offset. And so I can't quite tell who has a higher proportion, both or tumor only. Now, this stacked bar chart I got from one of the papers from your group, and I love this one. It's really good because I can see how the group composition changes over time, specifically looking at age of patients and the percentage of patients who either were in the surgical resection group, the contraindicated group, or the refused group. And I see that over time, as age increases, the percent of patients who are refusing and the percent of patients where surgery is contraindicated are growing. I love this plot. I love the palette that this plot is using. It's very high contrast, and I can easily distinguish the groups. And it's working so well because there's so many groups across such a large time that if the point of this plot is to show how those groups are changing, it's doing a great job. So waterfall plots are another kind of bar charts. Specifically, they're going to show treatment effect, and we saw this on our slide where we were looking at clinical research specific complex plots. And these bars that are showing treatment effect are going to be arranged according to their magnitude. How far off are they compared to the baseline? Because they're going to be compared to the baseline. So it's using at least, it might be using one variable, but it likely comes from two variables. One variable being the baseline and the other variable being another snapshot time after the baseline. It could be one variable if it is trying to, if the data that you're given already has taken the difference and has already done the comparison for you. You're also likely to have another variable that is categorical, because each bar typically corresponds to a different patient or sample. And this is going to be typically used to show patient response to a treatment, but the design can be used for other cases around the same. And so this is showing the change from baseline, and it's coloring the patients based off of the disease progression. And so anything that is below the baseline, the tumor has shrunk, and they're likely having very good responses, while anything above the baseline, the response is the tumor is not shrinking and there's not good response. Because of how waterfall plots are used in the field, there is an accepted notion of what above or below the baseline means. For other applications that are using a waterfall plot, but are not necessarily looking at tumors and how they grow, and they might be looking at something else, there is not this accepted notion of what above or below the baseline means. And that can be difficult for people. And so you'll have to be careful with your data. And we'll look at an example of this next. A major strength of waterfall plots is that they're displaying patient and sample heterogeneity within one snapshot. You can look at a lot of data in one plot. A weakness is that it's a snapshot that is comparing two time points. As we discussed, it's comparing the baseline and some point in time later. If you want to look over a range of time, you're going to need different alternatives like a spider plot or a swimmer plot. But a swimmer plot loses comparing that change from baseline. You could use a forest or a caterpillar plot to look at treatment effect, or a Kaplan-Meier curve to look at outcome. And you might even be able to like group your patients based off of the color categories we saw here. You could also potentially use a heat map to incorporate time, where each patient is a row on the y-axis and each column is a time point. And the color is used to show the change from baseline and arrange them according to response, or facet them according to response. So this waterfall plot example with the penguins data, I wanted to look at, for each penguin in the data set, how its body mass compared to the average, and I colored it by penguin sex. So each bar is penguin, And whether it's female or male controls whether it's purple or green. And if I look at this very quickly, I'm assuming that this means that females have a higher body mass compared to average because they're the positive numbers. However, this is misleading because my y-axis was computed by subtracting the actual from the average. So if I have a negative number, that means that it's higher than average. If I change how I am computing the y-axis value, then I'm doing actual minus average. And this makes more sense for someone who's looking at the plot briefly, that the males tend to have that higher body mass, and so they are above the line, while the females are below the line because they have lower than average. This is a waterfall plot example from one of your papers where it's using outcome classification, and this is a really good example of using waterfall plots outside of that tumor response. So I discussed heat maps. And what are heat maps? Now, full disclosure, I love heat maps, and so I'm actually going to try to make them sound less cool than they are because they're not something that they're cool, but they have their time and place. So it's a visualization of a table, likely using color. It can also be some sort of a map. It doesn't have to be a grid, but heat maps in a grid or table format is what is most common or known. They're mostly two-dimensional or a matrix with rows and columns. And they're always going to use color to display numbers. You might also display categories if you have a few of them, but mostly displaying numbers. You have categorical or numerical or a mixture, likely a mixture. And you're going to have at least 3 variables because of the two-dimensional structure. And these can be used for looking at a distribution across samples or change over time after an intervention or to show pairwise correlation for many groups. It's not recommended if you need to know or see actual values because they often have a lot of squares and so you can't always put readable values on them. But sometimes you can if you have few enough. And we'll see one where it does and you can read them. And they're often used together with hierarchical clustering, or if you want to reorder sample location to show trends or areas of similarity. So why would you use heat maps? What are their strengths? And their strengths are that they're giving a broad overview of a lot of data, and you can identify trends, areas of density, sparsity, or outliers. And they're very efficient and compact because they're simplifying all of this complex data into one visualization. The weakness of them is the matrix aspect can be difficult for viewers to grasp. So while I love them and they make sense to me, that doesn't mean that they'll make sense to everyone. And it can be difficult for people to grasp them. I would suggest trying a heat map if you have tried another plot and it just looks cluttered and you feel like you need more room and subplots aren't really helping you. A heat map might be a good way to go because maybe you need that extra dimension of data. Another weakness of heat maps is that when you're rearranging the data, which is often very helpful and can be done a lot, it can also be misleading to your audience if you haven't described how you did it. So alternatives for heat maps include a table, because if you have actual values or summary stats that you want to communicate, tables, it's already as similar to a table structure, table could be a way to go. A scatter plot can be used because it has that X and Y, that row and column. And you can use color or shape. You might use line plots with multiple lines. And if you need another dimension because heat maps aren't enough dimensions, a bubble plot can be used. Another option are parallel plots, but they're a little weirder and they would be like my last suggestion. I've linked all these alternatives so that you can look at them. But heat maps are good. So example for a heat map with a penguin dataset is really difficult because the penguin dataset is not that complex. So what I've done is on the rows, I've put the penguin sample ID and my columns are the sex of the penguin. So I also removed any of the extra like white space. So even though one row would seem like it's the same penguin, it's not. It's the penguin, the first penguin that's female, the first penguin that's male, the second penguin that's female, the second penguin that's male. And I've done the body mass example that we saw with the waterfall plot, where because I've ordered the data, we can clearly see that males have higher body masses than females do on average in this data set. So it's actually a really good quick way to do it, and we can see the range of values that all the penguins have. If I wanted to look at something like an average mean flipper length, if I have a lot of categories, this could be really useful. This example is less useful because I don't have a lot of categories. The Adelie are on 3 islands, but the Chinstrap and the Gentoo aren't. And the other problem that I have is that this corner up here with the Adelie male penguins is not very easily contrasted. Their values are really near each other. So the heat map is not the best example here. But another example of using heat maps are pairwise correlations. And so if I have multiple time points and multiple treatments, perhaps control with 3 different time points and treatments, sorry, 4 different time points with 4 different time points, I can look how my samples compare to each other. So control compared to control at 0 hour, control paired to control at 24 and 0 hour, and I can look at how well they're correlated. And so this is plotting the Pearson correlation for these, with the diagonal being the comparing to themselves, so they're perfectly correlated. Of note there, though, is that this is highly redundant because the top half is going to mirror image or be symmetrical to the bottom half. So for a pairwise correlation heat map, you can often just plot the lower triangle and you've reduced that complexity and you're still giving them all the information. And the diagonal is not included here because it's just assumed that these are perfectly correlated to each other. So looking at these, the stacked bar chart is good if you want to look at how subgroup composition changes at a glance over time, but it's less useful if you have a lot of subgroups. The waterfall plot is good if you have a baseline to compare to for a single time point, many samples in patients, and you clearly know what above or below that baseline means. And a heat map is good if you have at least 3 variables that you need to visualize and want to use color to show the range of values, the correlation, areas of densities, patterns across groups or time, etc. The final 2 plot types we're going to look at are more like graphics for tables or summarizing complex relationships. And the first of these is caterpillar plots. And caterpillar plots are going to look like a caterpillar when you look at them because they have numerical point estimates that are the caterpillar body. These might be odds ratios, coefficient effects, things like that, as well as their variation or confidence levels, which are going to be the legs of the caterpillar. This is practically the same as a forest plot, except that the categories are going to be ordered by the magnitude of the point estimates, which we'll see an example of. You're largely going to be using this with numerical data, but some categories, and you'll have several variables, and you'll want to find a ranking or used for meta-analysis to compare models or odds ratios. The strengths of these are that they're providing comparison or reference lines, such as a baseline or a null effect. They're readable pretty efficiently, and they're going to display intra and interstudy variation quite well. The weakness is that they can be cluttered and difficult to read if you have a lot of studies or models. And there's a lot of important context and information in the axis labels about where these estimates are coming from. There's also the fact that the plot is just showing effect estimate and uncertainty, but isn't providing any additional information such as bias, which a funnel plot could help you add in. So for this caterpillar plot example, I tried to predict the body mass of penguins by penguin species using the bill depth, the bill length, and the flipper length. And so I have different combinations of the predictor values and their coefficient estimates, and I've ordered them by magnitude. So the higher the magnitude, it's at the top, and the lower the magnitude, it's at the bottom. And I'm comparing it to that null effect of 0. This plot from one of your papers is almost a caterpillar plot. It's got all of that idea of the odds ratio and the variability. However, it's a forest plot because it's not arranging these according to magnitude. If it was arranging these according to magnitude, I believe this one would be at the top, the colorectal surgery for males, and that the cardiac surgery for females would be lowest at the bottom. Well, this example from your papers also is a caterpillar plot that has been rotated. So the observed expected ratio is now on the y-axis instead of on the x-axis, and the individual programs are on the x-axis instead of the y-axis. And this is showing many, 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 many more examples to the point where you don't know information about each of the individual programs. You can see the range of values, and you might be able to label one or two of the programs, but overall you're losing a lot of context. But this is a very good example of a caterpillar plot. Finally, Sankey diagrams. What are they? They're going to represent the flow or movement of quantities, e.g. patients or samples, through different categories, e.g. demographics or steps of an analysis or trial. And you're going to use these with categorical data, though you might have numerical counts rather than counting your categorical data. And you're going to have several variables. This is best when there's a change over time and you have some sort of longitudinal data or time points with categories at each time point. However, you do not have to have time points. An example is tracking patient response or symptom trajectories and patient cohort stratification. Their strength is that they're providing a lot of information about the study population and it's showing connected possible paths, and it's a visual graphical representation. The difficulty is that they are very complex to interpret, and some people just have a very difficult time interpreting them. So an alternative could be a table. You could also use a stacked bar chart, though you're going to lose the connection or path aspect of a Sankey diagram. Flowcharts and tree maps or some other custom-made visualization could work. It might also be very complex to generate a Sankey diagram. Not all software supports that. This is a really bad example of a Sankey diagram with the penguins data. You don't need to see the translations for this one. A table or a stacked grouped bar chart is going to work much better. I don't need to see the flow here at all. You could still track demographics that have been through a clinic or experienced some sort of intervention, or how many papers and how much funding went towards discovery for a certain demographic and how that's changed over the years. So demographics are still something that can be used with Sankey diagrams. It's just the data set I have is limited, and it's good to show examples where it just sometimes doesn't work. This Sankey diagram is showing patient cohorts, where at first it's showing the primary site. So where's the primary site of the tumor? Is it colorectal, lung, or breast? Where's the metastatic site for those same ones? And then where was their treatment of the metastatic biopsy? It doesn't have a strong time dependence, but the diagram works really well. In fact, there's no real time dependence to this. While the treatment obviously happens after knowing that the tumor exists, I don't have something like this follow-up or this year. I really like this diagram. This is another example of symptom trajectories where there is a strong time dependence. You have your baseline, you have your 4 to 6 week follow-up, your 3-month follow-up, and your 6-month follow-up. And how often are they experiencing that symptom? And then it shows the different paths or the different combinations of those symptoms and the people who were discontinued in the study. This is a great example of that. Similar example is this treatment trajectory, where it shows what is given as the first line treatment, what's given as the second line treatment, what's given as the third line treatment, so on and so forth. It does show when a patient experiences death, does not show when a patient discontinues and either doesn't need treatment or something. And you can see that here in this area of the second line treatment orange bar, there's a lot of white space that doesn't have gray coming from that orange bar. So they didn't die, but they aren't obviously marked on what happened to them. And so this is an example of, to strengthen this diagram, show what happened to those patients. Did they discontinue? Did they not need treatment? Something like that. So the strengths of these plots are that the caterpillar plot is good if you want to look at effects and variation in a glance to identify the best and the worst or see the range of effects. And a Sankey diagram is good if you want to look at the possible trajectories of symptoms, treatments, etc. And it's important to know what combinations happen or what the different possible paths are. There are so many more types of plots, and knowing which one is right for you takes a lot of work. You want to use the simplest or most mainstream plot you can. So if there's a plot that is the accepted plot in the field, that's what you're going to use. You want to make sure that the plot works with your data types, that you're using numerical and categorical data correctly. And you want to use design tools before you switch to a more complex plot. Are you able to use a facet or a subplot, dashed lines, text labels, colors, and shapes before you move on to a more complex plot? There are going to be strengths and weaknesses of every kind of plot. Every plot we talked about had strengths and weaknesses. And your plot choices may be field, context, and data dependent, but focus on one takeaway message. Some plots are better for exploratory analyses, like the rain cloud plots. And don't underestimate the power of a good table. Sometimes tables are the right way to go. Always compare the end plot to your data and make sure that they match, and follow those best visual design practices for ethical and accessible visualizations. Data visualization is a bit of an art, and it may be iterative. You're going to have to try different kinds of plots and build up your plot and make design changes to improve it. But always aim for understanding and readability over complexity. I provided a few links for places where you can practice, because practice is the key to making good plots. And I also have some resources that you can look at for all this information. Are there any questions?
Dude, I thought that was such a great talk. I can't even believe it. It was exactly what we were looking for, and I think it was like, I've put together talks. I know what you did to do this was a ton of work, and it's like so greatly appreciated because, man, was it helpful. I learned a ton. I'm incredibly grateful for that expertise that allowed you to make this, and for the clarity with which you presented it. Like, well, it's amazing. Really, very helpful. Are there any questions from the group? I don't think there are, because it was such a great talk. Like, I think I could spend the next like month going through your slides and following links. I love the data to viz like site. I was looking at that during your talk. I really think you fed us a ton, and I think we're all very grateful. I banged people over the head with logistic regression for an hour before you started, so I think they all need to take a break. But honestly, outstanding talk. Thank you so much.
Thank you. And if you have any questions, please feel free to email me. Let me add that to a slide. I'll add it to the resources slide. We're also going to be putting this plot into, or this presentation into a course soon that will be available through our website. I can email you when that's ready. Well, thank you all, and I think I have to go to my next meeting, so please let me know if there's anything I can do to answer questions or anything like that.
Great, thank you, Katie.
Thank you.
Thanks, bye.
Hey, everybody, I just signed off another Zoom. Sorry about that. Great, well.